channel?
Sounds good.
All right. We're starting. Okay. I'm just going to start off by saying hello to everyone who is viewing on our YouTube channel and welcome you to our second plenary talk of the day. So for those of you who weren't at the talk, my name is Alicia, and I'm one of the co-chairs this year for OA3C. And on this call with me, I have Megan, who's the other co-chair for OA3C this year, and our second plenary speaker, Dr. Sigal Balshine. So before we get going, I have notes for this talk. So if you have any questions for our speaker, you can add them to the chat bar on the right-hand side of the YouTube video, and then we'll be able to read them back at the end of the talk. And then I will also note that there is about a 10-second lag time between when we're speaking and when you guys see it on our YouTube channel. So if there's any kind of delay with us answering questions, that's why there's a little bit of a lag, just the nature of the YouTube live stream. So without further ado, I'm going to hand over the screen to Dr. Sigal Balshine from McMaster University for her talk this afternoon.
Thank you, Alicia. I want to just start off by thanking everybody who's showed up and is viewing this talk, and I especially want to thank the organizing committee, Alicia and Megan, and everyone on the organizing committee. It's a lot of work to organize a conference, and you guys have had to do it under these really weird circumstances. So like, hats off to you guys. This must have been really, really hard. I have a soft spot for this conference. I actually organized it back in 2003 with my lab under SARS. So SARS broke out just as we were putting on the OEC 3, and it's funny to kind of come full circle and come back to this meeting that I just love, because it's a really fantastic place for the ecology, evolutionary, and behavioral communities to come together and share their knowledge. So thank you for having me. It's a real pleasure and an honor. Thank you. Okay, so in the last few months, we've been bombarded by images like this one. It sort of doesn't stop. It's in the news, it's on the radio, it's in social media. We're thinking all the time about the crisis that's facing our planet. But for the next, I don't know, 40 minutes or so, I want you guys just to put this crisis out of your minds. I want you to push it out of your minds and think about yet another crisis that our world is dealing with, and that is the biodiversity crisis. So our planet is, as you will all well know, facing a biodiversity crisis where there's literally thousands of plant species, insect species, amphibians, mammals, birds, you name it, fishes that are faced with extinction. And there's a lot of different causes for this biodiversity crisis. The biggest and probably most important one is habitat fragmentation and destruction. But as I said, there are a bunch of other factors that contribute to this biodiversity crisis. Things like invasive species introductions, overharvesting, pollution, and climate change are all things that are contributing to the biodiversity crisis that our planet is facing. So what I'm going to do today is I'm going to tell you a little bit about some of the work that the folks in my lab and in my research team work on. And this is work that really looks at the intersection of what happens when invasive species and pollution collide. And so I'm going to spend some time talking first about invasive species and then about how they interact with pollution and why that matters. So my talk is going to follow the following outline. I'm going to first start talking about the species that I work on, the round goby, and its invasion into the Great Lakes of North America. And then I'm going to talk about the role that behavior has in the round goby invasion. I'm a behavioral ecologist, and I love behavior, so I'm going to tell you all about what we've discovered about its behavior and why that matters for invasion. And then I'm going to spend the third part of the talk talking about what happens when invasive species and pollution collide. So before we really get going, I think it's really important that we define terms. So I want to make sure that everybody knows exactly what I mean when I say an invasive species. And most of you will know, but this is an invasive species. This is the Asian carp. It's a species that's not supposed to be in North America, but has come and has spread in great numbers. And an invasive species is really any species that is non-native, that has spread and grown in numbers to the point where it becomes a nuisance for the way the ecosystem functions, the way humans interact with that ecosystem, or even for the economy. So it's not the case that all non-native species are invasive. It's only if they're really successful and spread to really large numbers and have an impact that we call them an invasive species. So why have invasive species been introduced so widely? Why are there so many? Well, what's happened is that we used to have traditional species boundaries that were really set by world biogeographical regions. And those are really much breaking down because of us, because of the way we move around the planet. So in the next slide here, you can see the planet, the different continents in gray, and you can see these bright red lines. And these red lines refer to shipping lanes. So these are basically shipping lanes between continents where goods are taken from one part of the world to another. And on the land, on the terrestrial part, you can see the light pink, which are basically these road networks. And you can see that most of our planet is covered with pathways for these invasive species. So we're moving these animals from one part of the globe to the other. And the damage they are inflicting can be massive, catastrophic even. This is a very tiny little insect, the mountain pine beetle, well-known invasive, and there are ecological, evolutionary, and huge economic consequences. This organism is devastating North American forests. In fact, the Great Britain's Nature Conservancy Council has tried to put a number on how much money is being spent on trying to remediate and prevent invasive species and the amount of economic losses due to these invasive species across the globe. And it easily reaches billions, if not trillions of dollars each year. So some estimates suggest that 5% of the world economy is somehow connected to these invasive species, either remediating or just plain old losses from the impacts of these things. So this is a cute little fish here, unfortunately, is an invasive species, and it's a species that I work on and have worked on for about 20 years. It's called the round goby, Neogobius melanostomus, and it's a little benthic fish. It lacks a swim bladder, and it sort of hops along the bottom, and it has a modified pelvic fin, so it sort of adheres like a sucker to the bottom and just sort of hops along. And this species has come to us from parts of Asia, from what's called the Ponto-Caspian region of Asia. And that's like the Caspian Sea, the Black Sea, and it's come with ballast water. So when transatlantic ships move their loads across continents, they often have to redistribute their loads by taking on water to balance things out. And when they take in water, they also take in these invasive species like the round goby. And so this species has come over from this area, not only to the Great Lakes of North America, but also it's now spreading like wildfire in Europe. It came about 20 years ago, but right now it's going through a major spread through Western Europe. And this species is a highly aggressive species. It outcompetes native species. It's not the first, and sadly, it's not the last invasive we're going to see, but there are some special reasons why we have to be concerned about this species. And that's because of the speed of its invasion. So it had a very rapid invasion in North America, and it's having an equally rapid invasion right now in Western Europe. So in 1990, it was first sighted in Lake St. Clair, a small lake off of Lake Erie. By 1993, it had colonized Lake Erie and Lake Michigan. The next year, it was found in Lake Huron. The year after, 1995, in Lake Superior. And in 1998, it was first sighted in Lake Ontario. So a very rapid spread for a very little fish that isn't supposed to be a very good swimmer. I study this fish mostly on the western edge of Lake Ontario in a place called Hamilton Harbour. So as I said, this rapid spread, one of the fastest aquatic invaders we've ever seen, really surprised scientists and really surprised conservation practitioners because nobody expected it. It lacks a swim bladder. It doesn't really move very much. It has a really small home range and it's extremely site faithful. So we've definitely helped move this species around, but it was a big surprise. So why is a species being so successful? Well, one reason is that it's highly tolerant. It can manage in all kinds of salt regimes. It can even live in brackish water, and some people suggest marine environments. It can tolerate all kinds of temperatures, really high temperatures, really low temperatures, and very low oxygen as well. It's an opportunistic forager. It will eat anything. It really likes to eat dracaenid mussels, zebra mussels, but it will eat the eggs and young of many recreational sport fish. And so a lot of people really hate it because it's having impacts on the very native species we want to see more of, that we recreationally love to fish. It spawns for a long time. It starts spawning in late April and it will keep on spawning till October. Most of the fish in our regions have a very short breeding season, maybe like 1 month, 3 weeks, something like that. But this fish can spawn many, many times in a year. They have male care. So the males look after the babies, and that means really high young survivalship. So this is another reason why it's so successful. And as I said a minute ago, it's really aggressive. It's a really good competitor. It outcompetes natives, and it's capable of living at quite high densities. So that's sort of thought to be some of the reasons for its success. So now I'm going to turn our attention to some of the research that my lab has been doing to try to ask if there's a role for behavior in the round goby invasion. And I guess I'm just going to start off by saying that there's sort of two contrasting behaviors that a lot of people attribute to invasive species. The first is aggression, and the second is sociality. And these seem like they're different sides of a coin, like complete opposites, if anything. And but yet there's research that suggests that both of these behavioral traits can help a species invade. So there's a bunch of evidence, a bunch of papers that have suggested that the individuals that move an invasion, that spread an invasion, are the aggressive, the bold, the competitive, the intolerant individuals in a population, because they don't like to hang around others and they'll move and disperse and get away from others. And so they move the populations and cause this range expansion. But there's also conflicting evidence in the literature that says, oh no, it's the least aggressive, the least competitive, the more peaceful and gregarious individuals that are actually contributing to the invasion success because they're kicked out of established areas. These are the least competitive individuals that have to be pushed out into no man's land because they can't hang on where there's established populations. So there were these conflicting papers in the literature, and my lab was really interested in knowing how behavior might contribute. Does behavior contribute a role in invasion biology? And so what we did, and when I say we, I really mean Miriam Groen, who was a third-year project student in my lab, was we asked the question, is aggression linked to dispersal and range expansion? And are individuals at invasion fronts more aggressive? Now, we were really lucky because we collaborated with Mike Fox, who was at Trent University. And Mike's research group had been watching round Goby spread in the Trent River system for some years. They knew that there had been an introduction at Hastings in 2003. And so we went to Hastings with Mike and his students, and we picked up gobies from this established area. But Mike had also been watching these fish move up the Otonabee River through Rice Lake and up the Otonabee River. And they knew that there were gobies that had just arrived that spring at a particular site. And so we went to that invasion front and picked up fish and compared them to the fish from that introduced spot that had been previously established 7-8 years previously. And so what we did when we got them into the lab is we matched them. We matched pairs. We had one fish from the invasion front matched with a fish from the established area. And we matched them in terms of size, because body size is really important when you have a contest. And we gave each of these fish a compartment, a little shelter. Gobies love shelter. They'll work really hard to stay in a shelter. They'll fight other fish to keep their shelters. We gave them each a shelter and we left them there overnight. And then the next morning, this is sort of what happened. So they habituated overnight to these new areas and they were each given a shelter. And the next morning, their shelters were removed, and so were the barriers that separated them. So then they could see each other, interact, and there was a shelter in the middle, and they had a contest over that shelter. And we videotaped the contest and all the behaviors that they performed. And we assessed shelter monopolization, who got the shelter after these 20 minutes. And then we checked again after an hour and a half, and then after 3 hours again. And then we stopped and separated the pair. So before I go on and show you some of the results, I have to tell you that Goby fights are really interesting because they occur in these two phases. First, you have this kind of assessment phase where there's no contact and they just sort of figure out how big the other one is. And they do this by slapping water against each other. They have these sensory mechanical cells called a lateral line down their sides, and they basically can feel how big their opponent is by these water slaps. They'll also perform these puff throats. They try to make themselves look as big as possible. They'll raise their fins. They even bark at each other and make these sound vocalizations, but they're really not touching each other at this non-contact display phase of the fight. And then after that part of the fight, they turn into an overt contact phase where they'll start chasing and biting each other. The guy with the pointed yellow arrow is about to bite the other guy. And the fights usually have bouts. So they'll have a display and then some overt aggression, a display and then some overt aggression, and they have a little break in between. And then finally it's over. There's a definite winner and a definite loser. And the winner just keeps on chasing the loser and the loser just keeps on running away. And you can really easily tell who's won and lost in most of these cases. So most of the time, there's also what we call hierarchy formation. So the winner is dominant and gets the shelter, gets the little house, it's totally happy, and the loser has to stay outside and runs away every time the winner comes near him. So what happened in our experiment? Well, we found that there was more fish from the invasive front that won contests. About 65% of the contests were won by fish at the invasion front. And they tended to perform more aggressive behaviors. But by far, the thing that predicted who won and who lost most of all was body size. So even though we tried so hard to match them up in size, even the smallest amount of body size differences, like they were less than 5 millimeters difference, the bigger fish would typically win. And when the invasive fish was even a tiny bit smaller, even by a couple of millimeters, it wouldn't necessarily win the fight. So we found kind of a mixture of evidence here. So it was the case that invasion fish tended to be more aggressive and they tended to win the shelter more often, but the difference in body size really dictated the outcomes of these fights. And we think this is much more likely to happen in nature as well. Well, what about the other side of the coin? What about social behavior? How is social behavior contributing to this invasion of this round goby, this little benthic fish? We know that round gobies can live at really high densities. People have estimated extremely high densities, many fish in a meter squared in various lakes. And we know from breeding behavior studies that they nest close together. They have breeding aggregations. So we thought sociality might play a role in their invasion success. So here we had there were two wonderful students who took on these research questions. They wanted to ask the question of, are round gobies attracted to conspecifics? And if they are attracted to conspecifics, do they prefer large aggregations of conspecifics to small aggregations of conspecifics? And if they do prefer conspecifics, do they prefer them more than they prefer shelter, which we know from other studies they strongly prefer? And the two wonderful students who did this research were Pauline Capel, who was a thesis student in my lab a few years back and now works for the Ministry of Natural Resources, and Erin McCallum, who was a PhD student in my lab and is now a postdoc in Sweden. So what did we do, or what did the girls do? The ladies went to Lake Ontario and used minnow traps to collect fish in the western basin of Lake Ontario. And we brought these fish into the lab and we had three different behavioral assays. In the first assay, we simply said, do fish prefer to be with another individual or on their own? Will they spend more time beside another individual or go to the empty side and stay away from others? In the second assay, we asked, will they go to groups of 3 versus just a single fish? And in the third assay, we asked, will they prefer to go to shelter than to hang out with conspecific, another goby? So really simple behavioral measures, and we had very simple predictions. We predicted that because gobies are sometimes taken by other fishes and by birds, that it might be safer to be with others, and that fish might prefer to be with conspecifics for predation reasons, for anti-predation reasons. And what we found was that both males and females in our study strongly preferred to be with near the fish rather than near the empty compartment. So they had a strong preference for others. Our second prediction was that they would, for the same reasons, prefer to be in large shoals than with just a single another individual. But here we were absolutely wrong. So when we compared 3 versus 1, we found that they had no preference for the large versus the small shoal. They just were going towards other individuals pretty much at random. And our third prediction was that we thought that the fish, because shelter is so important for them, both to avoid predators, but also to breed and mate and lay eggs and care for those eggs, we thought they'd show a much stronger preference for shelter over a conspecific. It's safe to be with another, but it's even safer to have a nice strong rock protecting you that you can defend from others. And here we found there were sex differences. So females had a strong preference for shelter as we predicted, but males actually showed no preference at all. They spent about half their time with conspecifics and half their time interacting with the shelter. So to summarize this section, round gobies are grouping together. We see that in nature, but they don't really seem to discriminate too much between large and small groups, or at least not groups of 3 versus 1. And their tolerance of conspecifics may have indeed facilitated Great Lake invasion. We need to do more work on this. We also see that males and females may value safety versus sociality somewhat differently. And this is work that we're continuing in the lab now to think why that might be. So I'm going to sort of pause for a minute now and tell you that I've been just saying males, 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 as if all males are the same. But we all know that's not true. And it's definitely not true of round goby males. So male round goby are not all the same. These are the picture of 2 reproductively capable males. They're both round goby. They both have large testes. They both can reproduce, but they look very different to us. And we come across this all the time. When we pull up our nets, when we're doing our research on round gobies, we see that they come in two different morphs. They have what we call alternative reproductive tactics. There's a sneaker male, and there's a garter male. So the garter male is usually black. He has secondary sexual characters. He has puffy cheeks. And the sneaker male looks much more like a female. He's almost like a female mimic. He's the size of a female or even a juvenile, but they both have turgid genitalia. So the white bars there refer to their genital papilla, where the sperm comes out. And you can see it's quite swollen and it looks the same size, and it really is bigger for their body size in sneakers. So they have like a fire hose, if you like, for a genital papilla. And this is research that my students and I have been doing together, and we've been looking at what are the differences between sneakers and garters for some time. So the person who started this research is Dr. Julie Marentet. She now works for the Department of Fisheries and Oceans, and she also gave an OEC talk when she was a student in my lab. And the first year that Julie decided to attack this question, she collected about 700 males, and about 300 of them were what we call reproductive males. And she found the darker morph, the were much larger than the lighter morphs. So there were these light mottled males that looked just like females, but they had large testes, and we had a much darker, puffy cheek, wider head male. And she called these garters and sneakers. And I'll tell you why in just a minute. But just remember right now that the garters are bigger than the sneakers. So when she opened them up and she dissected them, she found that indeed the garters, these black males that are the ones that look after the babies and court the females and defend nests, these dark males had large testes, but relative to their body size, they actually had smaller testes than the much smaller sneakers, which were basically just swimming bags of testes, because their testes were pretty much filling their entire body cavity from their anus all the way up to their throats. They were just full of testes. And so in absolute size, they weren't that different. But in terms of relative size, once you control for body size, the sneakers were investing much more in making testes. But gobies are neat because they also have these things called accessory glands on their testes. And in terms of accessory glands, it was actually the garter male, the parental morph, that had the bigger accessory glands. And we think accessory glands are really important for attracting females and maybe even for parental care purposes. And again, this is an active area of research in my group. So Julie also had these fish, she collected ejaculates from sneakers and from garters. She found that in the ejaculates, when she spun down these ejaculates, she found that the volume of sperm was much higher in the sneaker morphs than it was in the garter morphs. So for the same volume of ejaculates, the sneakers are putting out many more sperm. And she also did some hormone work to suggest that one of the big androgens for fish is something called 11-ketotestosterone, or 11-KT. And it's responsible for all those beautiful secondary sexual characters in fish that we see, the beautiful coloration and the longer fins and so on. And again, here it was the garter morph that was different from the sneaker morph. So there are these big differences in their physiology and in their morphology. So that's all well and good, but you kind of wonder, like, okay, are sneakers way more common? Are garters way more common? Who has the upper hand? What's going on in nature? And to do this, you sort of have to track the invasion. And my group has been watching round gobies in Hamilton Harbour, monitoring round gobies in Hamilton Harbour for 20 years. We go out every 2 weeks and we sample gobies with minnow traps. And we do this from pretty much the end of April until November. So we have a lot of different years of data. We have a lot of different sites, and we have a lot of gobies in our database. And if any of you ever want to look at this data, we'd be very happy to share and do all kinds of collaborative work. We have a very rich data set that we'd love other people to mine as well. So with that in mind, two of my grad students, Erin McCallum and Anish Bose, looked at about 9 years of data. And they took this data and looked at how many garters and sneakers were there in the population. And the dark blue lines here refer to the garter types, the big black males that look after young and defend a nest and court females. And the kind of hatched line that's sort of in a lighter blue is the sneakers. And the very lightest color of all, the gray, refers to males in the population that are non-reproductive. So they're not sneakers and they're not garters. And if you open them up, their testes are weedy and they have very little sperm and they're just not involved in reproduction at that time. So the first thing you can see is that initially in the beginning of the year and early in the season, the garters are about 2 times more common than the sneakers. But over the entire season and over the entire many years that this data refers to, it's actually the non-reproductive males that are the most common males in the population. Oh, that wasn't what I was expecting, but so what we have found so far and what I'm going to summarize right now is that darker garter males are larger. I just said that. They have wider heads. They have higher levels of a particular androgen, 11 ketotestosterone, and they're 2 times more common. The lighter sneaker males are smaller. They have larger testes relative to their body size. They have also narrower heads and they have much denser ejaculates. I want to emphasize that all our evidence so far, we can age these fish using ear bones called otoliths, suggests that they're actually of a similar age. So there's not really an age difference. It's not that sneakers grow into the garter as they grow bigger. That doesn't seem to be what's happening. And we've also done a bunch of diet studies as well, which I don't really have time to talk about right now, but happy to talk to anyone later who wants to talk about that. So that's all great, but I want to tell you that the round goby, because of it's such a prolific invader, and right now Europe is being bombarded by them. There's a lot of work being done on round gobies and their impacts. In fact, we recently did a review and found that since 2009, when we published our evidence for alternative reproductive tactics, there's actually been 333 different papers on round goby. That's a lot of research on round goby, but only 4% of them even mention that there's an alternative reproductive tactic or distinguish whether the males are reproductive or not and what kind of males they are. So we know that there's still a lot of work to be done to make the message clear to people that a fish isn't a fish isn't a fish. There's a lot of big differences between these animals. More specifically, no one's really looked at how the tactics might differ in their behaviors. And I told you I'm a behavioral ecologist, so I'm really interested in behavior, and I wanted to characterize how these are different. And lucky for me, I attracted a wonderful master's student, Katie Shinishin, who was also really excited to try to solve this question. So that's a picture of Katie. She's not going to be very happy for me showing you this funny picture, but she's been doing research in my lab, asking questions about round goby, alternative reproductive tactics, and their different behaviors. So what Katie's been doing is she's been looking at things like boldness, exploration, sociality, aggression, and even dispersal tendencies. So, and she's been comparing the different male morphs that she's been collecting in the field and running them through assays that allow us to get information on things like these differences in behavioral tendencies. So how does she do that? Well, we have something in our lab called the exit test. Lots of behavioral labs use the same test, but it's basically a start box with a trap door that we can open remotely. And we just look, does the fish want to stay in that shelter or will it come out and explore? And what we're finding is that sneakers are much more likely to explore than our garter males. So this on the y-axis here is exit time. And you can see that the sneakers are coming out of these shelters more quickly than our garters, but there's a lot of variance, obviously. Another assay that Katie has used is something called our exploration assay. And here all we do is we have a bunch of, we have an arena with grids and the fish move around a novel tank that they've never been in before, and we ask how much they explore it. And although the difference isn't huge, the sneakers tend to explore more of the maze or more of the arena, I should say, than do the garters. So they're more likely to explore. We also have something called a sociality index. And basically what we do here is what I just told you about a few minutes ago. We ask, does the goby want to hang out with 3 others that are just like it, or does it want to hang out with nothing, with just a shelter? So we have an asocial zone and a social zone and even a neutral zone. And we ask, where do the fish place themselves? And right now our data is suggesting that there's not really a big difference between garters and sneakers in where they place themselves. They both prefer to be with conspecifics over alone, but there's really no difference in that preference. However, what they do with those conspecifics does differ. The garters are much more likely to be aggressive and attack and bite and try to mouth fight against the other conspecifics, while the sneakers don't do any of that. They just stay nearby and are quite quiet and passive. And finally, we have this large flume that we call our dispersal assay. So we put fish in this start box and we film what they do. They have to go through a maze and through barriers to get to the end. And there's always a pump, and the gobies like to swim against flow. We also have these canyons and valleys. They have to swim through these kind of challenges or little barriers that they have to hop up and then go through a valley and then come up and go down and so on and so forth. And we asked, does one tactic reach the end more frequently or more quickly than the other? And the answer was no. Only about 45% of garters and only 38% of our sneakers actually ever reach the end. We made the dispersal chamber maybe a bit too hard for them, but there was no difference in the average time to reach the end of those that managed it. So to summarize, I've shown you that the garter males are definitely more aggressive. And I've also shown you that sneaker males are a bit more exploratory. They came out of a shelter and they're exploring a new tank more than the others, than the garter morph. But the tactics actually had very similar dispersal tendencies. So what I'm trying to argue here is that you should be thinking about round gobies just as you would orangutans. Orangutans, males, come in 2 forms. There are some males in the population that have big cheek pads and phalanges, big wide heads, just like the gobies. They make these calls, they court females, they perform consortship, they guard their females. And then there are other males that don't have any of those secondary sexual characters. They still reproduce, but they do so by using an alternative tactic. They basically try to intercept a female who's in a consortship with one of these cheeked males. And so I'm arguing that alternative reproductive tactics exist, and there are big behavioral differences between these males and even between reproductive males and non-reproductive males. And I think we need to think about these things. And I think that ecologists and evolutionary biologists and behaviorists don't think enough about these things. And the reason I think it matters is that if you have a bold individual or an exploratory individual, it's much more likely to get trapped. And we as scientists are constantly sampling animals and trying to decide something about the population based on our trap data. But I'm arguing that these behavioral differences are really going to influence the inferences we can make about the population, because there are going to be behavioral differences in who gets into a trap or who stays in a trap. So now I'm going to turn our attention to the third part of my talk, and I won't go too long. And if we're running out of time, please just start waving at me wildly. I want to talk about what happens when an invasive species and pollution collide and meet each other. So contaminants are a worldwide problem. It's not just in Canada, it's not just in North America, it's a worldwide problem. And at really high doses, we know that contaminants can be lethal. But there are lots of problems that can occur because of contaminants that happen at sublethal doses as well. And those are the things I'm really talking about, and the things that me and my students are really interested in asking. So we're particularly interested in the round goby as a vector for contaminants, because this is a fish that whenever it can will eat zebra mussels, will eat dracaenid mussels that are really, really good filter feeders. And these mussels have really cleaned up the water quality in the areas which they've invaded. And now the goby is a specialist on these mussels and can release these toxicants by allowing other animals to eat it. So things that were previously locked up in the mussel tissue, because these zebra mussels are such good filter feeders, are now available. They're bioavailable because the zebra mussel is one of the is a prey item for the round goby, and the round goby can eat these zebra mussels while few of our Great Lake fish can. But many things are now eating the goby, and so it's a vector that possibly can mobilize contaminants in food webs. And so we got very interested in this species for this reason. Also, I happen to be a professor at McMaster University, and for those of you who don't know, McMaster sits right on Lake Ontario. Our campus actually touches Lake Ontario. You can walk to Lake Ontario from campus. And Hamilton Harbour, which is the area that is closest to us, is an area of concern. The International Joint Commission between Canada and the US has identified Hamilton Harbour as an area of concern in the Great Lakes. All of the orange dots are areas of concern because of their contaminant loading. And there are a few green dots, which are good news stories. These are areas that have been delisted, that have been cleaned up. But there's a lot of remediation happening in Hamilton Harbour, and there's a lot of pollution in Hamilton Harbour. And that's mostly because historically there were no pollution laws. So between 1910 and 1970, the steel mills could do what they wanted, and a lot of deposits were put into Hamilton Harbour. And there are many contaminants that are considered a problem in Hamilton Harbour. I've listed some of these below, and the ones in red are ones that are beyond what Environment Canada says is safe. Safe levels. So there's a lot of stuff, a lot of it's in the sediment and not in the water column, but it gets stirred up every time a ship goes by. And they've recently capped the major coal tar deposit. So we're really excited to see if the harbor is really beginning to show even stronger signs of restoration. There's also a lot of sewage effluent that reaches the harbor. So we have something called sewage overflow sites, and these are combined sewer and drains from homes. And because Hamilton's an older city, we don't have necessarily a separate system for homes and businesses and sewers on the streets, and they get combined. And anytime there's an overflow and a really high rain event, raw sewage can end up entering the harbor because it can't make it. There's just overcapacity, and it doesn't make it to the 4 squares, which are the wastewater treatment plants around Hamilton Harbour. So we have wastewater treatment plants that are supposed to treat all this raw sewage, but under really heavy rain events, raw sewage can flow into the harbor. So there's multiple different kinds of contaminants happening in the harbor. And the yellow dots here just refer to the historical area of Hamilton Harbor, and whatever is being built on top of that is really the steel mills, and they've really built into the harbor itself. It's all artificial, but it's really this north end of Hamilton that has a lot of the industry. And similarly, the contamination in the sediment and even in the water is very heterogeneous. So there are areas where there's tons of contaminants in the water, really, really high levels of things like PAHs, and other areas where it's really low. And we make use of this heterogeneity in our studies. So we go to areas where it's relatively clean. There's not as much in the sediment or in the water column. And we compare that to areas which are really heavily impacted. And we fish there, we sample there, and we bring these fish back into the lab. And we track the invasion using very simple methods. So this is called a minnow trap. I don't know what's happening right now, but I'm just going to skip. I must have taken something that had an audio on it. I apologize. When we collect our fish, as my audio is just saying, it's using minnow traps, and we bait the traps. We also have unbaited traps, and we compare those. We collect data on how many fish were caught, their body sizes, their sex ratios, their reproductive status, and we also open them up and take them back to the lab because these are invasive fish and we're not allowed by law to put them back. And we take these fish from cleaner sites and from highly contaminated sites. And what we're finding is that in the cleaner sites, the fish are bigger than in the contaminated sites. And they typically also older. So there's fewer younger fish. Sorry, there's more younger fish in the contaminated sites and fewer older fish in the contaminated sites than in the clean sites. And you can see that over the years, the body size of these fish is also being reduced. We're also finding that when we examine their tissues, that there's higher levels of contaminants in the fish from these contaminated areas in the orange or in the gray compared to fish that we've collected from the cleaner areas in green or white. And we have all kinds of evidence that they're impacted. There's higher levels of metals in their livers. There's more fin erosion. So if you look at their pelvic fin, which is this important structure for them of how they stay put in flow, it's usually burnt off or highly degraded in contaminant sites compared to clean sites. And there's also a lot of evidence that there's some endocrine disruption. The males have a yolk protein called vitilogen that's expressed at really high levels, and usually it's just females that will have this expression of vitilogen. The males also have feminized genitalia. Their external genitalia is feminized. In the contaminated site, it looks more like a female genitalia on the outside. And if you open them up, you see more evidence of intersex. This is a male, but inside of the male's testes, you find eggs. So there's a lot of evidence that these fish from these highly impacted sites with high levels of sediment contaminants have been feminized and have endocrine disruption. Of course, because we're a behavioral lab, we're interested in what does that mean for behavior? What's going on behaviorally? So we put them in things like dispersal fumes and ask how long does it take clean fish versus contaminated fish to explore and move and reach the end of a dispersal flume. And what we find is that contaminated fish are less exploratory than were fish from clean sites. They explored later and they move a lot less. And if we scare them, if we pretend to be a predator and chase them, we can catch the contaminated fish more quickly, more easily, even if we're blinded to whether the fish came from a contaminated site or a clean site. And if we scare them with a robotic fish called robofish that we've had in the lab, we find that overall the contaminated fish are more sluggish. They are less active before anything happens, but after you scare them during the attack, they both freeze. But the fish that have come from contaminated areas return to their baseline activity much more quickly. So in the orange line, you can see the baseline for the contaminated fish was returned to much more quickly for those populations, while the other group did not reach baseline even after 15 minutes post-attack. So we've also done a bunch of contest situations and find that fish from highly impacted sites where there's a lot of contamination take much longer to sort out their dominance hierarchies or to win shelters. They'll switch who's dominant and who's subordinate much more fluidly than fish from cleaner sites, who seem to figure out who's dominant and who's subordinate much more quickly, who's the winner and who's the loser of a contest. Okay, so that's all great. We took a bunch of fish from dirty sites and we compared them to fish from relatively cleaner sites. But of course, we need controlled exposures. That's not good enough. So we've been doing a lot more of that and asking, how does exposure to contaminants for different lengths of time, for 3 days, for 21 days, for 28 days, for several months, influence behavior and physiology? But there's a lot of different behaviors to ask questions about. Which behavior should we choose? What should we do? There's so many different things we could choose to look at, and we've been looking at a lot of it. One of the behaviors we always try to look at is activity. Does it go up or down in response to these exposures in the lab? And we've also been choosing to focus a lot on aggression, because aggression is an important fitness-related behavior. It determines how much food you get, whether you get a shelter, whether you can maybe look after your young really well. And so it's a more complex behavior than activity, and we've chosen a kind of simple behavior and a more complex one to focus our attention on in the lab. So we've done a lot of aggression assays after these exposures, and also activity assays, asking how long it takes them to explore and move through a maze, and so on and so forth. There's a lot of different contaminants to expose fish to in the lab. What should we do? There's so many different things in the mixtures that is the real world that we really have to make some sort of decisions. What are we going to expose the fish to in the lab? And our lab so far has been really focused on two things: pharmaceuticals, which is a huge class of contaminants on its own, and what we call wastewater, because really when you take some drugs, often it's not completely metabolized by your body and it's entering the aquatic realm again, because many of our wastewater treatment plants aren't really completely prepared to get rid of these drugs. So we've been looking at real world wastewater, collecting wastewater from the wastewater treatment plants, treated wastewater, exposing fish to that, and also to various pharmaceuticals. And I don't have time to talk about all the different studies we've done. I'll just give you a little bit of a teaser and remind you that the reason we've looked at wastewater and pharmaceuticals is because the two for us are connected. Pharmaceuticals are getting into our waterways through treated wastewater, and the treatment not being completely good enough to take away a lot of these pharmaceuticals that we can find in detectable doses near wastewater treatment plants, and they are affecting animals. So here's a study that we've done in our lab. With wastewater exposure, we can expose them to a control or no wastewater, to a low dose, say 10%, or to a high dose, say 50% wastewater, treated wastewater effluent. So this is wastewater that's being completely treated and is entering our lakes and our rivers again. And what we're seeing is it depends what you're studying. So we've seen no change in Goby aggression with these kinds of treatments, but we see that at the highest dose, there is a decrease in activity, for example. And again, this is a sort of a message of it depends on what you're studying, because if we study the drug fluoxetine and give them various doses, we find that there's a big impact on aggression, but no impact on activity. So the two the simple behavior that we chose as an assay and the complex behavior that we chose as an assay are giving us different responses as an example. So to summarize this third part of the talk, I've told you that round gobies can and do live and thrive in very polluted areas like Hamilton Harbour. And I've told you that round gobies are smaller and slower, and they're not as good at getting away from predators when they're caught in naturally contaminated areas and tested in the lab. And I've told you that when we expose animals, round gobies, chronically to wastewater, it slows these gobies down. And exposures to particular drugs like fluoxetine can make the gobies less aggressive. So finally, I want to just say, end my talk with a huge thank you. And the first thank yous have to go to the two wonderful women who did all this contaminant work. Julie Marentet has been very interested in metals and inorganic contaminants, and Erin McCallum has been more interested in pharmaceuticals and wastewater and more organic things. But we've been connected, all of us, and interested in knowing how these contaminants influence behavior of round goby and other native fishes. And so I also want to thank the many, many collaborators who made this study possible, and the funding agencies that funded the work, and the many government and conservation organizations, and the City of Hamilton, and Environment Canada, and Department of Fisheries and Oceans, all being partners in the work that I've described today. And so finally, I am done, and I hope you've stayed with me. I know it was a bit of a blitzkrieg, and I'd be really happy to entertain questions through the moderators, but also if you want to write to me, email me. I'd love to have a Zoom call in these strange times. As Melissa Bateson said this morning, I'd love to have a beer with you at the poster session, but that's not possible. So please Zoom me and ask me any questions about the work that I described today in a rather rapid way. Thank you.
All right, I'm going to do a clap here since we don't have an audience, but thank you. We do have some questions coming in on the live stream right now. So we have a handful of them right now, and hopefully we'll get a few more, but I'm just going to start with the first one. So this one's actually from Aileen, who's one of our committee members, and she asks, during the preference tests, were the conspecifics presented were the conspecifics familiar or unfamiliar? And do you think that the familiarity would impact the preferences and the social behaviors?
That's an excellent question. So in our preference tests, the conspecifics were all unfamiliar. So we made sure that they did not know these fishes. They weren't from the same stock tank or holding tank. So that's another research question that we could definitely ask. We have not looked at the impact of familiarity on preferences, but it's certainly something that I have done with my cichlid work, which is another area of research another area of research that I work on, and we've not asked that question with gobies, but it's a great question. Thank you.
All right. So next question, this one's from Christina, and she asks, how do you age the fish?
So fish have a bunch of different ways to age them. None of them are terribly easy, but inside the fish's skull, there are these two little bones called otoliths. In fact, there are 3 bones on each side, but there are these bones called sagittal otoliths, and they have rings on them, much like a tree would. And the otolith sort of grows bigger with time, and each year there's usually an annual ring that's put down much like a tree. So if you know what you're doing, you can polish them and read these rings. And I think I even have a picture. I don't know if you guys can see that online, but these are what the bones look like. You can see there's a lot of variation in their shape. And we have found that although they're similar in age, the sneaker morph seems to have grown less by the time winters come. So the space between like how much it's grown from the center to the first ring is much less in sneakers than in garters. And that's a hint that maybe that first year of growth is really important in changing your trajectory to become a sneaker or to become a garter. But we really don't know. It's a big open mystery. But they sort of sit on either side of the brain, and you sort of have to go in there and get them. Other people can age fish from scales and even from the spines. You can take a spine off the dorsal fin and cut it. And again, you see those rings that grow. I've never done those other methods. I've never used those other methods, but they do exist and other people have used them and validated them.
All right, thank you. So the next question asks if there's a difference in the invasion zones in regard to the type of male. So is one of the phenotypes more invasive than the other?
Oh my God, whoever asked that question, please reach out to me because I have been trying to get my students to answer this question forever. We've been really wondering about this for ages, and every time we start a study, something happens, like there's a flood at the invasion zone, or there's a drought, or there's COVID, or. So this is something that I have been trying to answer for many years. I have been involved in some modeling projects that look at this from a theoretical perspective. And so if you reach out to me, I can tell you what the theory suggests. But mostly, I will really want to figure this out and haven't yet. So I'm desperate to figure that out.
Okay, perfect. We have a couple more questions, both related to the male phenotype. So this next one is from Gavin, who asks, are the male morphs condition dependent or genetically determined?
We don't know. Another $1 million question. The only hint we have that it might be condition dependent comes from what I'm looking at right now, which is the suggestion that sneakers actually have less first year growth than the garters, but that's the only hint we've ever had. We have no idea if a sneaker male has sneaker sons, and if a garter male has garter sons. We have no idea. And that's because gobies are really hard to breed in the lab. So we have never managed to grow up very many. It's really hard to get them to do anything like really mate in the lab. We can collect animals that are guarding, but they don't keep on doing it in the lab, or very, very rarely. Like we get a few a year, but not enough for a study.
All right.
It's really odd that there are some invasive species which are so hard to culture in a lab, yet they do so well. It's just mind-boggling.
I know. Isn't that bizarre? Yeah, I feel like we're missing some sort of magic bullet or a special ingredient to get them to thrive in our lab. And I should also say that while I haven't had that much success with it, there's been a bit of success in Europe, in a lab in Europe, in breeding them. And there's also been quite a bit of success in Linda Corcram's lab when she was at Windsor. And they had a really strong flow through system. And I have a feeling that that's one of the key elements. I think they need a lot of flow. And we don't have that in my system. We have a static system. So I might be like thinking about this all wrong, but I think it is a really interesting finding that other invasive species too don't thrive in a lab, but you throw them out and they're weeds. They grow everywhere.
Okay, yeah, we have a few more questions coming in. Most of them are related to the sneaker versus garter phenotype. We have one here from Hannah that's asking if females have a preference for one phenotype versus the other.
We do not know.
We don't know? Okay. All right, let's see, one more. Do you think that it's possible that both the garter and phenotype and sneaker phenotypes are needed for the breeding process?
No, I don't think so. I think it's absolutely the case that you need the garters for breeding. I don't think the eggs would survive for a second without a garter, but I don't think the sneakers do anything except for maybe deter the garters from staying around. So, in a really beautiful study by Brian Neff, who's at Western University, Brian's been able to show that having sneakers around really means that garter males in bluegill sunfish will care less than when there aren't sneakers around. So it may be that the garters can detect that they've been cuckolded, that their paternity has been stolen, and that they provide less parental care when there's a lot of sneakers around. But how that affects the carrying capacity of the population and the general growth of the population is something that I'm really interested in, like the alternative reproductive tactics. What does that do to invasion probability, and what does it do to the population's carrying capacity is, again, a really interesting question that I've worked on theoretically.
All right, I think we'll cut it off there. So thank you, Dr. Balshine, for answering all of these questions. So we had about 60 people or so attending the talk on our YouTube channel. So pretty good turnout, which is awesome. And we got our handful of questions. So for everyone who's watching, if you have any further follow-up questions, you can contact Dr. Balshine on or via her email. Do we have a slide with the email address on it? Maybe. No. Okay. Anyways, I will post the email address with the YouTube video. But yes, thank you everyone, and thank you, Dr. Balshine, for speaking to us today in this new kind of online format.
Yeah, thank you for having me. And just to everyone who's participating in the OEC, it's such a great meeting. And if you're an undergrad or a first year grad or just a junior grad, just know that this is the spot where a lot of us got our starts. And it's a really wonderful venue. And thank you for the organizing committee for taking this on and doing it. It's really a graduate student trainee conference for trainees. And it's really odd to have that and really wonderful. Well done to you guys.
Applause. All right, I'm just going to end the stream. All right.